So to facilitate discussion, I have candy bars.
All right.
And this is just not a candy bar. This is a Three Musketeers candy bar. Okay? So the rules are anybody who volunteers a question gets a candy bar. Anybody who asks answers a question gets a candy bar. Okay, so let's get talking, and if you have any questions, just bring them up. Okay, I don't get paid by the slide, so if we don't get through them, it's okay. All right, we can talk about everything else later. So part of growing any plant is dealing with diseases. There's very few plants that don't have many diseases. Roses, because of their diversity, run the whole gamut from many of them not having diseases to some of them being magnets, frankly. So we're going to talk about, the whole idea is going to be focused on trying to head off disease. Because once you get a disease, you get behind. Because we have growth curves, and I don't know, is this irritating more? Is it? Okay, let's take it. All right. Okay.
I'm answering that question.
You know, I can tell that, there's always somebody in the audience who gets more than their fair share, and, they might end up with a root canal afterwards, but I really appreciate them, right? So anyway, yes, we're going to talk about how to think about diseases so that you can head the problem off, okay? You want to head it off. You don't want to let it get going. So, if I can figure this out.
Have you ever used one of those before?
The problem is I probably used about 60 of them before.
That's the question.
And they're all different.
Yes, he's used one. There's the answer.
All right, now, Texas got our quarterback. And so you probably heard that, well, the Dallas Cowboys got our quarterback. So you probably heard that we're in the cellar because we don't have a quarterback. So I'm in training.
Oh, there you go.
Oh, yeah! All right.
Just throw to a Longhorn.
All right, so to think about it, we'll take something from the Bible. And it's just for every disease, there's a cause and a season and a host. Now, when you think about the whole thing, that really just kind of summarizes everything we're going to be talking about today. All right? Just in a nutshell. And so the way we think about it is, let's start with the plant. Let's just start with a rose. Okay? And it's hard to generalize on roses because, just as we heard earlier today from Ping Lin, there's a huge and, anyway, there's a large diversity of species, right? Large diversity. And then they're all hybridized together, and we just have a whole spectrum of rose plants. And because we have that, each plant in our garden is almost unique. You see what I mean? Each rose is almost different from the rose next to it. And so what I would encourage you to do is to start a garden journal. And so you keep a page for each rose in your garden, and you then have an overall app from like the Weather Underground. So you copy the weather from the Weather Underground into there, and you have those two things together. And the first year, you're going to curse me for wasting your time. Guarantee you. This is the biggest waste of time. Why am I even doing it? The second year, you're going to start to see a pattern start to emerge. Okay? The third year, you're going to be able to start to make predictions. Okay? So you start, it just takes time. Okay? But you'll start to see the patterns. So just one of the common things that people always ask is, why is the yellow rose so more, in general, susceptible to black spot? Well, according to the Consulting Rosarian Manual, it's because of the susceptible genes from Rosa. How do you pronounce it? Fotida? Fotida?
Petita
Okay. And, you know, that's just an example of the diversity in our gardens. Okay? So then you have to have a cause. Okay, now the cause can be living or it can be non-living. Okay, so in September, what was it, September 20th, we had Talk Like a Pirate Day. Right? You've heard Talk Like a Pirate Day. It's an international day where people kind of walk around like they have a peg leg. And they say, "Arr, you scurvy dogs." All right? And so what are rose hips good for? Scurvy. Scurvy. See, she gets the candy bar. Somebody over here gets the candy bar. Was it you? Oh, okay. Yeah. So scurvy is a disease, but it's caused by a nutrient. So it has a cause, but it's not a non-living, it's a non-living cause, not a living cause. So you can have diseases that are caused by abiotic, antibiotic factors. Now, if they're caused by a living factor, okay, we call that living, such as fungi, bacteria, and viruses, right? Then it becomes a numbers game. Okay? And it's just simple numbers. So when I walk up and I shake this gentleman's hand who's falling asleep right here, so I can wake him up, right? And he's just coughed into it. What's happened?
Uh-oh, you got the germ.
I've got a germ on my hand, right? Okay. All right. He's contaminated me. And so then I'll rub my eye. What happens? That's right. Now that germ has moved into my mucous membrane, and it has started to infect me. Do I get sick right away? No, that's right. Who said no?
She said no again.
Okay, and somebody over here said no too. All right, so I don't get sick right away. It takes some days, right? Anywhere from 7 to 10 days depending on the virus. Okay, so what's going on during that 7 to 10 days?
They're multiplying.
Multiplying like crazy. Absolutely like crazy. And so it's a numbers game, and that's what I mean about numbers. And so if everything is really good in the environment, you start out at a really low level, and it grows very, very quickly. It doesn't grow like this or like this, but like that.
Exponential.
Exponentially is the term. I like to try to stay away from these scientific terms.
That's mathematics. That's science.
I'm from Mississippi. What can I say? Okay? So what happens then is that if you don't take care of a disease early, you're taking care of it right up here. And the chances are that you're going to fail. Just like if you wait too long to go and get that antibiotic for a bacterial disease, say a sinus infection, you're going to be, 20 days underneath the weather with that sinus infection instead of like a week. Okay? And so you head it off right down here. Now, the importance of this is really extreme, but you'll never, if you do it right, you're never, ever going to know that you did it right. You just won't ever know it. And so you're going to be thinking, oh, I'm so tired, I don't have time to do this. Well, 30 days, 40 days from that time, you may pay for it. So let's give you an example. You go into the doctor's office, what's the first thing the doctor does? Come on, candy bar. What's that?
Take your temperature.
He takes your temperature. Actually, he should do something before that first.
How you feel?
He asks how you feel, that's right, or she.
Credit card.
But what's the first physical activity that physician should do?
Insurance.
What's that? Wage? Wait. Okay. Yeah, they're taking your parameters, but the first thing that doctor is supposed to do now
Ask you how you feel.
is wash their hands.
Oh, yeah.
That's for insurance.
Okay? Wash their hands. That is sanitation. They are removing the bacteria and fungi from their hands so that it doesn't transfer from the previous patient to you. Okay? And studies have shown that when the doctors wash their hands, the transmission within a medical facility is a lot less. Right? So, washing your hands, basic sanitation in the garden, is going to pay you back big time. And the sooner you act on a disease problem, the more you've reduced the numbers, and the numbers make the disease. Okay? So, now we have a plant, which is the rose, huge diversity of what is in a rose. And we have a cause, and that cause can be living or non-living. Okay? So just put these two together. You've all seen this. And in tolerant rose varieties, the Blackspot, you have just this general kind of netting. And we know this is Blackspot because what? The edges of the lesions all have this kind of feathery look to them. Right? That's like the key of recognizing Blackspot. And so there's a set of genes in the fungus that then interact with the set of genes in the plant. This is also Blackspot, but it's manifesting differently because of the different fungus, the different hosts, but you still know that it's Blackspot because you have the fuzzy edges. But the plant is starting to react to it more severely because you're starting to see this chlorotic severe reaction around the lesion. And then this is also Blackspot, but a highly susceptible variety. Again, that reaction between the set of genes in the fungi and the set of genes in the plants are causing this specific reaction. Okay? And so this is a really susceptible rose variety. All right. Now, talking a little bit about Blackspot, putting it into context. There's numerous races of Blackspot, somewhere between 5 and 12, depending on who you want to argue with. Okay, maybe even more. Okay, current research that there indicates there's going to be more in the future. About 8 interaction types. 5 compatible, 3 incompatible exist between the plant and the black spot. So you've got a lot of stuff going on in the garden. And then this is really one of the puzzles that I think is cool as a geek, right? But takes a bit to think about. The diversity of black spot across North America is not large. Okay? It's really not. But the diversity within a big garden can be significant. Huh? What does that mean? Why is that?
Got a lot of variety of plants.
You've got a lot of variety of plants, you're right. Okay?
Kind of a sugar high.
But also, yeah, you'll be able to make a good Three Musketeers smoothie. And so what's going on is that we ship plants all over the country. So the black spot from Georgia or Mississippi ends up in Arizona or California, and the black spot coming out of the Pacific Northwest comes over here and comes from down south and from California up to the north. So we have this huge mixture going on because of UPS and FedEx. We can blame them, can't we? Okay. All right. So that means that all that diversity of black spot is mixed up, but you don't necessarily buy all your plants from all over the country. You may get your plants more locally, so you may have 2 or 3 types or races of black spot in your garden, okay? But not the 5 or the 8 or all the interaction types. And so this explains, and you've probably all heard it, that I've had somebody who's just getting into plants has this old garden rose that's been beautiful for years. But supposedly it's really susceptible to black spot. And you don't know why that has escaped. Well, if that person starts bringing in more roses, then more of those races come in, and probably within 3 years, that plant which has survived 50 years is all of a sudden going to be diseased and weak. Okay, do you follow what's going on? Okay, and does it make, question, see? Candy bar, good. The question was very good, and actually there were 2 of them. So changes, current research indicates changes in the future, and does that mean that there's going to be more races, and are those races maybe indicated to overuse of a fungicide and fungicide resistance? Did I encapsulate that? Okay. So what's going on is that we really haven't figured out yet what all the resistance factors are and the genes that are interacting in the rows between the fungus. We've got a lot yet to learn. But what we're seeing as we start to look at the DNA sequencing and things like that is that it looks like we're underestimating the diversity that's there. We don't know how to measure it yet, but it doesn't look like we're sufficiently getting that diversity. Okay? And so, does that then relate to the fungicide? Probably not. We're looking at the genetics and not so much the fungicidal resistance. Okay? It's kind of 2 separate things that kind of relates but not wholly relates. Okay? It's kind of like E. coli resistance to a bacteria. Thank you, antibiotic. Who said that? Candy bar. So yeah, it's kind of like that. Sir.
Okay. Well, talking about black spot. Okay, will they, I want my candy.
He's getting hangry, isn't he? He's getting hangry.
Okay, if I can answer. When you spray your roses with a like propofol, will that take care of all kind of black spot, or does different kind of black spots require different kind of fungicide?
Right. So the question was, if you're using one type of fungicide religiously to spray your roses, will that handle all races of black spot, or will it, or will it not? Basically, is that right? And so the answer is, it kind of depends. And usually the answer is yes, it will handle all races. But you may have, say, a fungicide that's not particularly effective. It's kind of effective, but not greatly effective. Then maybe one of those races of the black spot can start to increase in prominence in the garden, and then that one can start to overwhelm the system. So you're actually selecting against the other fungi, and that one is replacing it. So that's kind of what I mean.
Excuse me, but you're talking about sanitation.
Yes, ma'am.
Do you consider like using spray as sanitation, or how do you know when to start? Like that you've got black spot.
Right.
Maybe you don't, and so how do you know? Like when the first sign of black spot, when do you know that?
Okay. So really good questions. So let me see if I can encapsulate them. Correct me if I'm wrong, if I missed something. So when do you know when? Let's see. Does a fungicide act as sanitation? And when should you implement sanitation
Well, yeah,
practices?
when do I know my first sign of black spot, or do I before I even see black spot?
Right. So should you do it before you even see black spot, or once you see it? And my answer is once you start to see some diseases, such as like downy mildew, you're already behind the 8 ball. Okay? And so part of why I like you to keep that garden journal is so that you can look into your crystal ball and say, I think this weather pattern is going to be conducive to this disease, and then you can spray proactively. Because fungicides work a lot better when they're used prior to the onset of the actual pathogen and the disease. Okay?
Now, what if you can't? I don't want any more candy because this is still my question. But what if you can't? Like I live in Canada, I can't get fungicides anymore. Can I use milk? Can I use
Sure, we can
like that?
we can talk about that, and I'm going to use Blackspot as a model to show you kind of how to think about things a little bit.
All right, thank you.
Okay. So yes, sir.
I have a question. I have black spot, of course, with the feathered edges, but I seem to have some black spots now that are very well defined. Is that?
Yeah, we're going to go ahead and we're going to look. So the question was, he has black spot with the feathered edges, but now he also has some spots that have very defined edges. And part of what I want to do today is just kind of take a survey of the common ones we see in the South. This isn't going to help you in Canada. Okay? It's not. Okay. Yes, ma'am.
In North Texas, you best spray as soon as you plant.
Yeah,
If not, before we get to the nursery.
right. And we're going to talk about exactly some of that reasoning. Okay?
Question.
Yes,
If
ma'am.
you do spots, you got a little, does it help to pull those leaves off? Does it lessen the,
We're going to talk about that. Yes, ma'am.
you can throw it.
Oh.
Always, yes, that question.
All right.
So, just about saying, you don't answer.
All right. See, I want to go play for the football team so I can make some money, right?
You don't run out of candy pretty soon. We'll never get through this talk.
All right, so we've had a cause, and it can be living or non-living, and then we have the plant, which has that huge diversity we call rosa. Okay? And then we have an environment. Because if we don't have an environment, we probably not going to have disease, right? Everything's perfectly controlled, okay? So there's a whole bunch of things that go on in the environment that are critical for the development of disease, and these are just a few of them. But look at the predominance of free water and humidity. It's pretty darn close to over like 50% of the pie. So this is, if you're an accountant or a political scientist, you probably always heard the phrase, follow the money, follow the money, follow the money. Well, if you want to start trying to figure out where a disease gets started and when, follow the free water and the humidity. All right? Free water, rain, irrigation, dew, and those are the major ones. But we also have some things called guttation. Most of you know what guttation is? No. Okay.
No.
So you have a rain, soil saturated. You have really high humidity in the air. You walk out in the morning and you see all these beautiful jewel-like gazing balls coming off the tips of every rose leaf in your garden. Okay? That is guttation. And what has happened is that the water pressure in the soil has pushed into the roots, and the evaporation off the leaves has stopped because there's so much humidity in the air and there's no sunlight. So that water pressure pushing on the roots is pushing the water up through the stem of the rose, out to the leaves, and then out these little water holes called hydothodes at the tip of the leaf. Okay? And so as that water comes out, it's come through the leaf, and it's bringing all these nutrients and minerals with it. Okay? And so you have all these bacteria and fungi that say, free lunch, and boom, there they come. Okay? So, free water is like the place that you as the accountant need to start. Okay? So, let's just kind of look at this. This is a soybean leaf. Sorry, I cheated. Okay? But look where the water is sitting. Can somebody describe that distribution of water for me? It's on the veins.
On the veins.
There you go.
I don't need any more candy.
Sure. Okay. All right. I just was trying to keep you up tonight. I didn't want to watch you. All right. So it's on the veins. And so you have probably all seen this in your garden. That is coming where that free water has set on those leaves. And where you got a little bend in the leaf and the water sits, that's where you're often going to get a lesion forming. Okay? So, we have three components. We have the plant and all its diversity. We have the pathogen or the cause, and we have the environment. And where they overlap, right in here, where everything is just perfect, then you get the disease. And we call that the disease triangle. And so any idea now why I'm handing out 3 Musketeers and not Snickers or Hershey's?
You got this.
You have to be 3 together.
That's right. You have to be 3 together. Okay? So while you're putting your garden journal together and you're thinking about this stuff, I want you to think about the 3 Musketeers, the cause, the environment, and the plant. Okay? Because they all work together. Sir?
Dr. Henn, you know what that is? It's a beautiful Venn
Isn't it a great Venn diagram?
diagram.
And that's 2 ends, just like a
That's
10, right?
H-E-N-N.
H-E-N-N,
I
yeah.
figure somehow you may have been kin to him and your name got changed. A comment, because I was at a workshop down there, I think it was probably maybe at Mississippi State, and it was most likely one of your professors. And what I'm seeing here, you're saying basically the cause has to do with free water and the humidity.
Many things can be explained by that.
But
Yes.
he gave something that I have kept, and I have actually taught this in doing programs, that when the combination of temperature and humidity reaches 160, your plants are very susceptible. So that when you, like in Arkansas, and the temperature's 90 and the humidity's 90, you got a good chance. You better be afraid.
Okay, where I'm going to argue with him.
Well, I'm not. It's not
Okay,
me.
I'm not arguing with you. I'm just going to say where I'm going to argue with him is that when you start to deal with a number like that, you're dealing with an extreme. And when you get into extremes, you're getting into plant stress.
Right.
And once you get into stress, then all sorts of things can happen that just depends on what's there to jump on. Okay?
Okay.
So what we're going to talk about as Blackspot as a model in just a minute is going to go totally against that. Okay? I mean, it will not 100%, but there's enough there to show that it's not necessarily
Did you
true.
go back and find out who in your department?
That's right. So sometimes in your environment, there's an overwhelming driver. And sometimes it's the environment. So like this year in Houston, you had huge rains, right? You had floods and everything, and that just overwhelms the system, and you probably had lots of sick plants later on in the summer, even into now, right? So that environmental variable just overwhelmed the system, okay? Similarly, you can have the plant overwhelm the system. So Dr. Ping Lim can breed a super resistant plant that even though the conditions are conducive, it's not going to get the disease. Okay? That's a doable thing. On the other hand, you can have one of these disease magnets that just, you know, if you turn around, it's going to have something, right? You know, they're just so susceptible that that's what it is. And then finally, you can have such a severe pathogen that no matter what you do, you're going to lose. Okay? It can just happen that way. So an example would be Ebola virus in humans. Okay? It just overwhelms the system. Okay? So we're putting all that together into that disease triangle. So let's use Blackspot as a model and just think about something that you know from your garden experience, have some comfort with. Okay? And we'll try to put some of this together. So you start out with this lesion, and that chlorosis grows and grows, and what happens to that leaf? It falls off to the ground. I got to get rid of all these candy bars. You can't say no. All right. That works. Okay, so it falls down to the ground. So here you have a bunch of leaves on the ground. And if it goes unchecked, the pattern of the fall of these leaves is what? Where does it first start in the spring? Where does black start?
New growth.
The bottom of the plant, the new growth. Who said that? And somebody over here said that, the new growth. Now, where are these fallen leaves? At the bottom. So what's happening is that you have, let me go one more, is that you have the pathogen overwintering in the leaves. The early spring rains splash that up to the lower leaves. It infects those lower leaves. They produce spores. Those spores get splashed up here. They infect. They produce spores. They get splashed up here. And the cycle continues till at the end of the season you have this long naked cane with this tuft of leaves and a flower at the top. Right?
Right.
You head to my garden.
So this is a really, really common pattern that we see in pathology. This pattern you can see in soybeans, you see it in peanuts, you see it in hydrangeas, you see it in all sorts of plants. And it's the overwintering of this inoculum that is causing this pattern to be exhibited. Okay? So how are you going to break this pattern? Sanitation. Bingo. So you're going to clean it up, rake and destroy, and let's see, who said sanitation? I'm sorry. All right. So sanitation, over here. So sanitation, and by doing it immediately, you are acting way down here rather than way up here. Okay? Now, this is not as simple of a deal as you might think. Okay? You want to dispose of the debris in a sealed container. One of these bags works great. Okay? You get done, tie it up, put it in the trash can. But what kind of trash cans do you buy now? Plastic. Who said that? Yeah.
Smarty pants.
All right. So you're buying plastic trash cans, and you've all experienced this. You walk out and you put your hand on that trash can handle, and you pull up that lid, and it's going. So you've just pulled a suction on that trash can. And if you just threw those leaves into that trash can and they've been sitting there for a day, you've just pulled all those spores out. And if you're upwind of your garden, you just did a good inoculation. Okay? That's what I'm saying. It's not quite as simple as it seems. You got to think about it just a little bit. Okay? What are you going to do with your disease material? How are you going to handle it? Where's it going to be stored until you can get rid of it? All those questions are really good questions that you need to think about. Okay? So, why worry about a little black spot? Well, yeah, it's unattractive. But eventually, as you all know, you get reduced blooms, you get, weakened plants, and maybe they're more susceptibility to winter kill and all this stuff. And yes, it will infect all roses, even knockouts, but not quite as badly. And so this is why we're all depending on people like Henn to get us better plants. Okay? And hopefully the CRISPR revolution is coming and will help us even faster. Okay? So let's look at black spot at the plant level. Okay? This is my mock-up of the leaf surface. And then this is the waxy cuticle, the thing that really shines and everything. And then this down here are just the cells in the upper part of the leaf. Okay? So then you have a spore, diplocarpin, rosy, 2 carps, all right, lands on the leaf. And it's going to land right there on the top of that leaf. And then water comes. And that spore has to be exposed to water for at least 7 hours. Okay, at least 7 hours. Right? For germination and the formation of the infection peg, and then finally for infection to occur, 9 to 18 hours. Okay? But if you have 100% humidity, so these are the days, those of us who live in the deep south, you all know these days. You walk out of your garage and onto that concrete floor and you slip, right? That's when that humidity is right up there. When that humidity is that high, only 5 minutes of water are necessary for the germination. Okay, so that was a really good question. It is, how long does it take for the infection to express? Okay, that whole period is called the latent period. The period between infection and expression is called the latent period. Okay? And we're going to get right to that in just a second, I promise. And so what happens then is that it germinates, and when it germinates, it starts using the material inside that spore to grow and make the infection. And this is a key point where some of the fungicides actually target, okay, this actual point in the development of the disease, okay? And so then it forms a germ tube, an apressorium and a germ tube, which then infects the plant, and then it starts and sets up housekeeping right underneath that waxy layer, but above the leaf cells, which is why there are times where you look at that rose leaf, you see that black spot infection, and you could swear you could scrape that thing off. Right? But that's why you can't. It's just underneath that waxy area. And then what it's doing is it's sending down Haustoria to feed on the cells beneath it. And these things kind of suck the energy out of the cells like a soda straw. Okay? So, the next stage is that you have sporulation and that you have the spores produce. Okay? And they, in a normal kind of moderate temperature, will take somewhere from about 11 to 30 days to produce that sporulation. Okay? Now, we're going to get to the weasel words really quick, the fine contract language. Okay? These canidia spread by rain, physical means like people, cultivation, insects, and then weeds. Now, let's look at the cause. Okay? All fungal isolates do not cause the same degree of infection, and they have different degrees of aggressiveness. Well, what does that mean? Well, it just means how fast that disease is going to occur. Okay? Probably there are several to a few different types of black spot in your average garden. Fungal isolates differ in their temperature preference. So an isolate from this area introduced into this area might have cooler temperature preferences, and this one might have warmer temperature preferences. Okay, so you have a diversity that's going on in your garden. And then you have all these varying symptoms in the black spot that we kind of looked at earlier. Now, let's look at the plant end of it. Okay? So if you look at it, what we've talked about already kind of comes out. Low stress level, less infection. High plant stress level, more infection. Okay? Young leaves, 6 to 14 days old, are much more susceptible than leaves which have stopped growing. Much more susceptible. So these are the small kind of reddish looking leaves growing out. Okay? Those are really susceptible. Okay? So if you look at the environment part of this whole thing then, what you see is that the spores themselves, depending on the isolate, will germinate from 32 degrees Fahrenheit up to 91 degrees Fahrenheit. Okay? The rate of germination depends on the amount of time exposed to free water and high humidity. Like, remember that 5-minute type thing. Infection at low temperatures occur. So infection can occur at 32 degrees Fahrenheit. But that latent period is significantly extended so that the expression of the disease doesn't occur until it's about 50. Okay? So you've had this infection, but you don't see symptoms until much later. All of a sudden now you've got whole new tools and ways of thinking about how to handle black spot in your garden. We've got 2 questions, so here and over here. First here.
Why are the younger leaves more susceptible?
Okay, why are the younger leaves more susceptible? The answer is, I don't know. There has been a lot of work done on waxes and lipids of the waxy leaves of roses, and a lot of people think that it has to do with a mechanical process. Immaturity of the leaf, not fully being formed, and so mechanically it's easier for the black spot to infect. It's all speculation at this point. Okay? Ma'am?
So the high temperatures work the same way, correct?
The high temperatures work the same way. Yes, ma'am. Just slowly.
Well, we don't spray in the summertime often in Dallas.
Right.
Because it's hot.
Right.
And there's no black spot.
Right.
But as soon as it cools off.
You get it.
Yeah.
Yeah. So yes, you've made a really valid point of looking at it. So what happens is that you've noticed at both ends you have slower infection, slower expression. So slower at the cool end, slower at the hot end. Okay? And so yeah, when it's really warm out, you can get away without doing a lot of management. But then when the weather person starts talking about a cool snap coming through, you better be out there ahead of time. Okay? All right? So, yeah, that's all good. Now, pH germination for these spores is 3.6 to 6.6 with an optimum of 3.8 to 5.4. What's one of our most common older fungicides? Mancozeb is an old one. What else was, somebody said something else up here? Sorry? Trifurin? Calcium bicarbonate. Absolutely. What does calcium bicarbonate do? Part of what the speculation is that it's reducing the pH on the leaf surface so that the fungus the fungal growth is impaired. Okay? And it's not working against the spore, but it seems to actually be working against the fungal growth. Now, whether it's only due to a pH thing or a chemical interaction, we don't know. Okay? But it probably has something to do with that pH. Okay, so new leaves, most black spot susceptible. So I don't care if it's 35 out, if those leaves are out, you need to be putting a barrier on them. Okay? You need to have some sort of fungicide protecting them. Now, I suffer from blinders at times. I get focused and my thoughts are just on something and I'll walk right away and forget something that I should have done. And in this case, I had a box of pamphlets talking about different fungicides in my office floor where I would trip over them when I walked out the door. Somehow I didn't trip over them. Okay, so I didn't bring that box of pamphlets. So we're going to have to talk a little bit about fungicides at this stage of immaturity. Don't use the DMIs. Anything that ends with azole as the active ingredient, do not use at before or up to this stage. The reason is that that whole group of fungicides is a growth regulator. Okay? And so I had been experimenting with applying fungicides during the warm periods, during the winter. Okay? And it does you do need to do that to keep your plants healthy. But don't use this group of fungicides because I had cool weather, warmed up into the 50s, and I sprayed some of the plants that I all sprayed with propiconazole remained green stems until the end of July. And then I lost them between the balance of trying to keep them watered and not too wet, right? So, these DMIs early on, don't use them. Wait until you get just a little bit more growth than this before you start applying them. So what are you going to use? Well, the thiophanate methyls, the strobilurins, all of that group of chemistry becomes good. Okay? Or just straight dithane, mancozeb, as we talked about earlier. Good. Okay? Even as cool as it is, chlorothalonil is probably going to work and not give you any burns. Okay? So, new leaves are most susceptible. Start applications as soon as possible. If leaf growth is beginning, do not use. Okay, we covered that, right? Okay. So now let's kind of switch from black spot over to other ones. But before we do that, do you kind of see what I'm getting at about thinking about the environment, the cause, and the host? Is it kind of coming together a little bit for you? Good, I'm seeing positive. Okay, so we're going to talk about common foliar problems in the deep south area. Sorry, no Canada. Some of this stuff is
We
there.
get all this in Canada.
All right, so let's talk about powdery mildew. There's a couple different types of forms of powdery mildew that you'll see. Some of them make these little nice round colonies, and some of them just evenly disperse throughout the whole thing. Now, powdery mildew is the exception to the free water rule. Everything has an exception. Okay? And the exception to the free water rule is powdery mildew, because powdery mildew likes humidity but not free water. Okay? So high humidity favors powdery mildew. And you can even wash powdery mildew off with the garden hose if the plants dry really quickly. Okay? Or the Cornell Rose formula, which is basically a teaspoon of baking soda and a gallon of water with a spreader sticker added. Teaspoon of a spreader sticker works really well. Tablespoon of each. Sorry. And you just got to keep it shaken up. All right? And that works fairly well unless it's more than a moderately susceptible plant. At least here in the south it does. Okay, life cycle of powdery mildew. And I got to ask a question. Candy bar in it. Is this font, the colors, is it too hard to read?
Blue is.
Okay.
Okay.
Okay. All right. So I got to change that. I was trying to keep them with, well, you see what I was trying to do, the host and the environment. Okay. All right. So high relative humidity, 42 to 80 degrees, but less than 90 degrees for the most part. Optimal's usually around 71. Spores are wind-borne for the most part, and they germinate within 3 to 48 hours, okay, after landing on the host. The fungus overwinters in the host and in the buds. And if you see these small dark spots in there, those are the hard overwintering structures that then come out and, cause the sexual recombination for the next year. This is what's happening with powdery mildew. You have the plant cuticle right here, and the cells underneath it, and then you have the fungus on top, which sends the Haustoria into the cells, where it then slurps the cell contents out. Okay, now think about this in a bud, where it likes to hang out. So you've got this new leaf that's all rolled up really tightly in a bud. And all those cells are really, really small yet. Okay, because they're in a bud, they haven't expanded. And so you have this powdery mildew slurping the water and nutrients out of some of those cells, and some of them are going to die. And when that bud opens up and those cells start to expand, the dead ones are not going to expand. And you get that. You get all that distortion and the wrinkling and all that obnoxious looking stuff. Okay? So that's kind of powdery mildew. Right? Any questions on powdery? Okay, almost any of the fungicides that you want to use for Blackspot are going to be effective against powdery. Although the DMIs are more effective against powdery mildew than the others. So the AZOLE ending ones are probably in general more effective than the others that you're going to get.
They killed the new growths.
The comment was they kill the new growth. I've never had that problem.
Did you say that a while ago?
Well, only
that the
Only when the plant is just starting to leaf out, put on the new leaves for the year.
But that
Once the
powdery
plant-
mildew,
Once the plant has got fully growing, you got some mature leaves, you're fine.
oh, okay.
Okay? It's just when the leaves are just starting to come out of the naked cane in the winter. Okay. All right, so management, improve air circulation so you have reduced humidity in the canopy. All right? Make sure that the sun meets, what is it, the 6 hours minimum of full sunlight is being met. And again, I kind of like the triazoles generally keep things a little cleaner than these, but under moderately susceptible plants, that Cornell Rose formula is surprisingly effective. Okay, botrytis. This seems to be more of a problem in the West than we have it, except for here we have a lot of bud blight. In the West, where they are using Dr. Huey wood stocks, root stocks and things, you can get it growing on the root stocks. And so when you bring that in and you open that container up, you'll see botrytis blight just starting to rot the roots. Okay? For the most part, what we have in the South is this bud blight. It gets its start on dead organic matter. Okay? And when it gets started, it produces thousands of spores. And then it can jump to things like this unopened bud. I'm out of time. 5 minutes. Okay. Then it may attack canes and pruning wounds and stuff like that. Okay? I'm just going to kind of skip through that because I'm going to skip through this because this is kind of rare, but this may be that circular one that you were talking, someone over here was talking about. Okay, so notice in this one there's no lesion. This is spot anthracnose. And this is the other circular one, which is a lot more common. This is Cercospora leaf spot. Okay, and yes, knockouts are not susceptible to black spot, but there's a place up in North Mississippi they cannot grow knockouts because of this. Okay? And this is the Cercospora. The way that you identify it with this $3.50 hand lens with the LED on the bottom of it, okay, is that you look towards the base of the lesions and you look for this black stuff coming up all from a central point, like a pincushion. Okay? So it's going to look kind of like a sea anemone. All right?
Yeah.
Okay? And so you have these perfect round spots with the gray center, and that's kind of what it looks like a little closer up. Okay? Same fungicides that work for the most part against black spot work here. Pain infections, I want to skip these to right here. Okay? So this is anthracnose or Colletotricum dieback. This rates like a one-sentence description in most places, but this is really devastating in areas of the Deep South. It has 2 forms. This is the leaf infection. Okay, the lesions are bigger, but they're round and they have a large kind of purplish cast to them. If you look on the inside of that leaf, you see these kind of roundy-like areas that are kind of blobby looking. Okay, versus that kind of sea urchin type look. Okay, this is kind of roundy, blobby. This is the cane dieback. Okay, and this is the cane dieback. Okay, and so what you have is you have an infection that happens somewhere way the heck up here, and it runs down with the upper part of the cane starting to die, and then this is the leading edge right here. You can see it's starting to penetrate the main stem. It's going to run right down there and kill that plant right there at the crown. Okay? It'll do this in just a few days. Yes, sir.
I have a question.
I got a question here.
Yes. Go ahead.
Could that problem be caused by damage from insects such as we're having right now from chili thrips?
So the question is, can this problem be exacerbated by insect or other damage such as from chili thrips? And the answer is, I don't know about chili thrips, but certainly from any other sort of damage. I've associated it with pruning damage. I've associated it with golf ball damage because of roses next to golf courses. And, you know, so yes.
Hail could do it, couldn't it?
Hail could absolutely do it. Yes. So this thing is really been important in the last few years, and the reason I want to ask you if you think you have this, I'd like you to send me a sample. Okay? And I want to confirm that that's what it is, and then I want to maybe try to sequence it and make sure that we're not dealing with several different types of fungi here and not just what I think it is. Yes?
But dealing with a problem in San Diego County is very similar to that.
I'm sorry?
We've been dealing with a problem in San Diego County that is very similar to that.
Okay.
Right? That's why I just took a picture of it. What environmental conditions favor that?
All right. Everything that I have on this is all my observations and are probably not 100% correct. Okay? This is what the cane looks like in dry weather. The blobs right here.
Yes.
That's after it's been a little more hydrated by free water. And you see how uneven these whole blobby things are? There's not a concise form. Okay? That is the key from separating this from the other cane diseases. Okay? And so what you want to look for are these asymmetric fruiting bodies. This is that same thing, and this is where they're starting to fruit. You can't really see it in this image, but those are almost salmon colored. Okay? And cool, moist springs, moist periods. I had associated this with winters, moist springs following winters with cool, warm, cool, warm, cool, warm type temperature fluctuations. Now I'm getting calls from a bunch of people all around, and it's happening in the fall. I don't know what's going on. This is why I want your samples to figure out if we're dealing with multiple things or what. I don't know. Okay, yes, ma'am.
Can you have some of that, what you think might be it, and still have a flower?
Yes, ma'am.
Maybe rose at the top?
Yes, but usually it'll kill the cane underneath it
Yeah.
that is growing it. Yes, ma'am. You sure can. Because what will happen is it doesn't necessarily start at the tip of the cane. It can start on a leaf and run down that leaflet midrib into the side cane, and from the side cane down into the main cane. Okay, it can come in anywhere in that whole architecture of the plant. Okay, I'm really out of time.
Oh, wait, what should you do?
Okay, fair question. Candy bar? Candy bar. All right, so first of all, spray the wounds with a penetrant fungicide. For now, I would not recommend using a DMI. I think it would work, but I don't know for sure. I have been using thiophanate methyl, and I have been drenching thiophanate methyl so that the roots pick it up and move it into the cane. So immediately you prune.
and you prune into healthy wood. If you prune and wood in where you have pruned is discolored, you need to wash and disinfest your pruners. And then prune again. Okay? Strobilurin will probably work, something like Heritage, but I've been using thiophanate methyl, and it's worked pretty darn well for me so far. Okay. 1 minute.
No, wash and sterilize your pruners how?
Okay. There's multiple ways to disinfect pruners. But what you have to remember is that anything you use to sterilize them has a limit of the amount of organic matter that it can tolerate before it no longer works. So wash the pruners so that they're free of organic matter, and then put them into the disinfectant. If you're cheap and you're using inexpensive shears, then you can use 1 part Clorox to 9 parts water. That's called a 1 in 10 solution. Okay? And you need to keep it in there about 3 minutes. Okay?
Between cuts?
I'm sorry.
Between cuts?
If you've contaminated, when you cut into that plant and you see brown stem in there, then you need to clean it and disinfect. Right. But if you have expensive pruning shears, you don't want to use Clorox. Okay? So I think it's
We used to use rubbing alcohol.
rubbing alcohol will work 70%. Don't use 100. Okay? But you gotta swab it down. The other thing you can use is this like MD20, and there's 2 groups that sell it now. And what you do is you look for the active ingredients, and it's going to start with N-alkyl, A-L-K-Y-L, and then it's going to go into this huge chemical name that's about this long. Okay? And that is food service safe. Okay? And you just need to keep your pruners in there for roughly 3 to 10 minutes. Okay? All right. Don't let the organic matter build up. You got to wash it first. Okay. So I'm sorry we just didn't get to downy mildews or the other canes. And okay. So
Come back next week.
Yes, I appreciate your patience. Thank you.